Hi everyone, thank you for taking the time to be here today. I'm Clinton Yam, a breast medical oncologist from the University of Texas MD Anderson Cancer Center. I would like to thank the Scientific Program Committee for giving me the opportunity to discuss 5 very interesting abstracts centered around novel approaches to target triple-negative breast cancer. Here are my disclosures. In the first poster, Lehman and colleagues from Vanderbilt University Medical Center, the University of Miami, and Baylor College of Medicine utilized a multi-omic approach to characterize triple-negative breast cancer to identify subtype-specific therapeutic vulnerabilities. As you are aware, triple-negative breast cancer is a collection of biologically diverse cancers with at least 4 distinct tumor-intrinsic transcriptional subtypes. Based on this knowledge, the authors hypothesized that subtype-specific vulnerabilities will be elucidated by an integrated multi-omics analysis. Using publicly available gene expression datasets, the authors found that the mesenchymal subtype of triple-negative breast cancer had lower stromal and immune cell estimates and decreased expression of immune cell markers, immune checkpoints, and genes involved in antigen presentation. Since DNA alterations can contribute to transcriptional diversity, the authors examined DNA mutations and copy number alterations within the 4 triple-negative breast cancer subtypes and found that mesenchymal tumors had a significantly higher percentage of epigenetic modifier mutations and were significantly enriched in ASXL gene family mutations that regulate the polycomb repressive complex 2, or PRC2. Together, these findings suggest that PRC2 activity could be increased in mesenchymal triple-negative breast cancer. The enrichment of epigenetic modifier mutations in the mesenchymal triple-negative breast cancer subtype led the authors to examine the DNA methylation patterns across the 4 triple-negative breast cancer subtypes. Of note, mesenchymal tumors displayed the most hypomethylated CpGs. Although mesenchymal tumors displayed global hypomethylation, there were distinct regions of DNA hypermethylation and concordant decreased gene expression in regions encompassing interferon gamma-induced genes, antigen processing and presentation genes, and EZH2 target genes. Given the lower expression of genes related to antigen presentation in mesenchymal triple-negative breast cancer, the authors evaluated MHC class I expression in triple-negative breast cancer cell lines with immunoblotting, immunohistochemistry, and flow cytometry, and found that mesenchymal triple-negative breast cancer cell lines had lower levels of MHC class I expression compared to non-mesenchymal triple-negative breast cancer cell lines. Since EZH2 has been identified as a catalytic subunit of PRC2, which has been shown to regulate expression of immune-related genes, including MHC class I molecules, the authors asked if PRC2 inhibition alters expression of immune signaling genes and MHC class I molecules. Indeed, PRC2 inhibition resulted in increased expression of immune signaling genes and MHC class I molecules in mesenchymal triple-negative breast cancer. Interestingly, PRC2 inhibition had limited effects on triple-negative breast cancer cell viability, and none of the 3 PRC2 inhibitors tested reduced viability by more than 50%. Despite the lack of in vitro efficacy, PRC2 inhibition increased intratumoral CD3 positive T-cell infiltration and increased the efficacy of paclitaxel in a 4T1 syngeneic xenograft model. In summary, the authors demonstrate through an integrated multi-omics analysis that mesenchymal triple-negative breast cancers have low expression of genes associated with anti-tumor immunity. Mesenchymal triple-negative breast cancers also demonstrated global hypomethylation with regions of hypermethylation encompassing immune signaling, antigen presentation, and EZH2 target genes. Treatment with an EZH2 inhibitor increased expression of immune signaling genes, but had limited effects on mesenchymal triple-negative breast cancer cell viability in vitro. Using an immunocompetent mouse model of triple-negative breast cancer, the authors demonstrated that EZH2 inhibition increased intratumoral T-cell infiltration and augmented the efficacy of chemotherapy in vivo. Taken together, these data suggest that EZH2 may be a key suppressor of anti-tumor immunity in mesenchymal triple-negative breast cancer by repressing expression of genes associated with immune signaling and those associated with antigen processing and presentation. Interestingly, in prostate cancer models, EZH2 inhibition activated interferon-stimulated genes and potentiated response to immune checkpoint blockade. Given the data presented in this abstract, it would be important to further investigate the role of EZH2 inhibition in sensitizing triple-negative breast cancer to immune checkpoint blockade. In addition to transcriptional suppression through histone modifications, emerging data suggests that EZH2 can alter oncogenic activity through methylation of protein targets, an example of which will be presented in the next abstract. Building on the role of EZH2 in triple-negative breast cancer, Gonzalez and colleagues from the University of Michigan identified EZH2 as an epigenetic regulator of p38, which promotes breast cancer progression. As you may know, EZH2 is a lysine methyltransferase that suppresses transcription of pro-differentiation genes. As we alluded to earlier, the most well-defined role of EZH2 is the trimethylation of histone H3, leading to transcriptional suppression of target genes. The p38 MAP kinase pathway is known to promote breast cancer invasion and metastases, and emerging data suggests that EZH2 interacts with p38 and increases p38 MAP kinase pathway activity. Based on these data, the authors hypothesized that EZH2 methylates p38 and promotes the malignant phenotype in breast cancer. In matched metastatic and primary triple-negative breast cancer from 16 patients, the authors demonstrated that co-expression of phosphorylated EZH2 and phosphorylated p38 was more prevalent in metastatic samples compared to matched primary samples. They further demonstrate that depletion of EZH2 reduces expression of phosphorylated p38, and re-expression of wild-type but not a phosphodeficient mutant of EZH2 restores expression of phosphorylated p38. Taken together, these data suggest that phosphorylation of EZH2 at T367 is required for p38 phosphorylation and activation. By immunoprecipitating p38, they demonstrate that re-expression of wild-type EZH2, but not the phosphodeficient mutant, is associated with increased methylation of p38. The authors performed liquid chromatography and mass spectrometry analyses of methylated proteins and identified K139 and K165 as unique sites of p38 methylation. Mutation of one or both methylation sites effectively suppressed methylation of p38. Using a pulse chain stability assay, the authors demonstrate that methylation mutants of p38 were less stable and had lower capacity for invasion compared to wild-type p38. Together, these data suggest that methylation of p38 enhances protein stability and promotes cellular invasion. The authors demonstrated that pharmacological inhibition of EZH2 and p38 suppresses various neoplastic processes such as proliferation and invasion, and promotes anti-neoplastic processes such as apoptosis and cellular differentiation. Notably, combined inhibition of EZH2 and p38 achieved greater anti-neoplastic effects compared to either agent alone. In murine models, the authors demonstrate that pharmacological inhibition of EZH2 and p38 suppresses tumor growth and development of metastases. In summary, these data suggest that EZH2 phosphorylation at T367 promotes methylation of p38, which enhances protein stability and oncogenic potential. Dual targeting of EZH2 and p38 reduces oncogenic functions of tumor cells. It is interesting to note that despite being in the same vertical axis, combining an EZH2 and p38 inhibitor resulted in greater suppression of tumor growth compared to either agent alone, suggesting that parallel networks involving EZH2 or p38 need to be investigated in future mechanistic studies. Building on the theme of epigenetic regulation in triple-negative breast cancer, The third poster, presented by Taro and colleagues from the Moffitt Cancer Center, describes a novel strategy of dual epigenetic and autophagy inhibition to target triple-negative breast cancer. MYC is an important driver of triple-negative breast cancer that is regulated by bromodomain proteins. However, pharmacological inhibition of bromodomain proteins has limited effectiveness due to the rapid development of resistance. Autophagy is an important mechanism of tumor survival and has been implicated as a key mechanism of resistance to bromodomain inhibitors. In this study, the authors seek to target the ULK complex, an early initiator of autophagy, in order to overcome resistance to bromodomain proteins. On the basis of these data, the authors hypothesized that dual targeting of bromodomain proteins and the ULK complex will result in enhanced anti-tumor activity. By examining ULK expression in publicly available datasets, the authors demonstrate that ULK3 expression was higher in invasive breast cancer compared to normal breast tissue, and high ULK3 expression was associated with shorter survival. In contrast, expression of ULK1 appeared to be lower in invasive breast cancer compared to normal breast tissue, and high ULK1 expression was associated with longer survival. These data are interesting because ULK1 and ULK2 are isoforms that are more widely recognized as autophagy initiators, while the role of ULK3 in autophagy is less clear. Of note, there are currently no drugs that effectively inhibit ULK3. Using breast cancer cell lines, the authors were able to further demonstrate that ULK3 expression was associated with the triple-negative phenotype and chemotherapy resistance. Consistent with its role in regulating autophagy, depletion of ULK3 resulted in a rapid decrease in downstream regulators in some 159 triple-negative breast cancer cells. As orthogonal validation, the authors were able to show that depletion of ULK3 impairs autophagy in WM1366 melanoma cells. In this model system, autophagy results in degradation of the GFP signal. From this experiment, it is apparent that cells depleted of ULK3 continue to maintain a strong GFP signal, suggesting that ULK3 is necessary for autophagy. SG3014 and its derivative MA9060 are dual inhibitors of BRD4 and ULK3. Treatment with SG3014 suppresses MYC expression in a dose-dependent manner in some 159 triple-negative breast cancer cells. Triple-negative breast cancer cells display greater sensitivity to SG3014 and MA9060 compared to JQ1. In contrast, the MCF7 cell line demonstrates sensitivity of similar orders of magnitude to all three compounds. Of note, the SUM159R cell line, which is resistant to both JQ1 and doxorubicin, is sensitive to SG3014 and MA9060. In an autophagy-dependent cell line, U266, treatment with SG3014 but not JQ1 inhibits autophagy. In summary, Taro and colleagues present in vitro data in support of dual pharmacological targeting of bromodomain proteins and autophagy with SG3014. Further studies are needed to characterize the in vivo efficacy of this novel compound. Our next abstract represents a collaborative effort between Houston Methodist Cancer Center, Texas A&M University Health Science Center, and the University of Texas MD Anderson Cancer Center. In this poster, Reddy and colleagues present data suggesting that inducible nitric oxide synthase activates PI3 kinase and AKT signaling in triple-negative breast cancer. As you may know, increased expression of inducible nitric oxide synthase is a poor prognostic indicator and associated with worse overall survival in triple-negative breast cancer. Nitric oxide regulates several oncogenic pathways, including the PI3 kinase pathway via P10S nitrosylation. PI3 kinase pathway activation is a known oncogenic driver in triple-negative breast cancer. Based on these data, the authors hypothesized that nitric oxide activates the PI3 kinase pathway in triple-negative breast cancer via P10S nitrosylation, and targeting the nitric oxide PI3 kinase axis suppresses tumor growth. Using proteomic data from the TCGA, the authors demonstrated that phospho-AKT expression is associated with iNOS expression in PTEN intact but not PTEN deficient triple-negative breast cancer. Additionally, co-expression of iNOS and phospho-AKT is more prevalent in triple-negative breast cancer cell lines and patient-derived xenografts when compared with those from other breast cancer subtypes. The authors further demonstrated that exogenous nitric oxide was able to induce AKT phosphorylation in a dose-dependent manner in triple-negative breast cancer cell lines with intact PTEN expression, but not in those with PTEN loss. In the PTEN intact SUM159 triple-negative breast cancer cell line, pharmacological inhibition of PI3 kinase was able to suppress nitric oxide-induced AKT phosphorylation, suggesting that nitric oxide is an upstream regulator of the PI3 kinase pathway. Notably, dual inhibition of iNOS and PI3 kinase suppressed tumor growth in PTEN intact patient-derived xenografts regardless of PIK3CA mutation status. In a phase 1-2 clinical trial evaluating the iNOS inhibitor LNMMA in combination with chemotherapy, iNOS inhibition demonstrated promising signs of clinical activity in patients with chemo-refractory triple-negative breast cancer. In summary, Reddy and colleagues present data suggesting that iNOS may interact with PTEN under nitrosative stress conditions and inactivate PTEN function. As demonstrated through early phase clinical trials, iNOS inhibition may be an effective therapeutic strategy in chemotherapy-resistant triple-negative breast cancer, which can potentially be synergistic with PI3 kinase inhibitors in patients with PTEN intact tumors. Further studies are needed to better define the molecular mechanisms underlying these data. Given its proposed role in regulating the PI3 kinase pathway, it would be interesting to investigate the implications of iNOS inhibition on the immune microenvironment in immune-competent murine models, as well as in primary human tissue from ongoing and future clinical trials of LNMMA. In the final poster, Sato and colleagues from the Icahn School of Medicine at Mount Sinai in New York sought to characterize changes in the tumor immune microenvironment in response to a novel cancer stem cell inhibitor. As you are aware, cancer stem cells are associated with resistance to chemotherapy and immune evasion. Inhibition of cancer stem cells has the potential to enhance sensitivity to chemotherapy and/or immunotherapy. Prior attempts at targeting cancer stem cells have focused on single pathways or molecules. However, pathway redundancy and plasticity of cancer stem cells limit the effectiveness of such approaches. Thus, the authors hypothesized that simultaneously inhibiting multiple cancer stem cell pathways will augment anti-tumor immunity and enhance response to therapy. 108600 is a novel cancer stem cell inhibitor that was identified through a phenotypic screen for inhibition of CD44 high and CD24 low triple-negative breast cancer cells. Of note, 108600 demonstrated potent inhibition of specific kinases implicated in stem cell maintenance. In immunodeficient PDX mouse models, 108600 sensitizes drug-resistant triple-negative breast cancer to chemotherapy. In further studies, Sato and colleagues treated mice bearing E0771 tumors with 108600 or DMSO for 3 days. Tumors were harvested for bulk RNA-seq. Gene set enrichment analysis revealed that treatment with 108600 resulted in downregulation of T regulatory signatures. To validate their findings, tumor-infiltrating lymphocytes were isolated from harvested tumors and analyzed by flow cytometry. Significant findings in E0771 tumors included a reduction in T regulatory cells and myeloid-derived suppressor cells with 108600 treatment. In 41 tumors, treatment with 108600 was associated with a statistically significant increase in cytotoxic T lymphocytes. Notably, in the E0771 model, 108600 treatment was associated with a reduction in PD-L1 expression in tumor cells. In summary, the authors provide interesting preclinical data suggesting that the cancer stem cell inhibitor 108600 sensitizes drug-resistant triple-negative breast cancer to chemotherapy in PDX mouse models and modulates the immune microenvironment in triple-negative breast cancer. However, several questions remain. First, are there data demonstrating in vivo efficacy of 108600 in immunocompetent mouse models of triple-negative breast cancer? Second, 108600 seemed to affect specific tumor-infiltrating immune cell populations in the E0771 and 4T1 models in different ways. What are the authors' thoughts regarding the underlying reasons for this observation? Finally, what is the impact of 108600 treatment on PD-L1 expression on tumor-infiltrating immune cells? I would like to congratulate all authors for developing this impressive body of work in triple-negative breast cancer and express my appreciation to the Scientific Program Committee for organizing this session. Last but certainly most importantly, on behalf of all the investigators, I would like to express our heartfelt thanks to every patient who courageously volunteered to participate in these studies. With that, I thank you for your time and hope you enjoy the rest of the program.